All right. Welcome back, everybody. Hope everybody had a good Easter break. And let's get going with our final stretch of this class. Okay. So we're going to keep talking about the reproductive system. When we left off, we had started out thinking about how different reproductive tracts develop. And then we had moved into talking about the typical male reproductive tract and some of its structures, including the development of the male gametes, the sperm. Okay. So in order to get our minds back in gear, I want you to take a look back at that and think about that sperm development pathway we were looking at between our Sertoli cells. And take a look and let me know which one of the cells that we're producing along this line leading us to sperm are able to undergo mitosis. Because as we're thinking about today and then moving into the female reproductive tract, we're going to be thinking about mitosis and meiosis and why it matters which one of those two processes is doing. Look and go over this. All right, cool. So spermatogonia is the right answer. And let's take a look at what we're talking about here. Okay, so what we're talking about when we're talking about sperm development was formerly called spermatogenesis. So the development of fully mature sperm eventually. So we're looking at some of the cell types that we had focused in on in the seminiferous tubules in the testes. So here are Sertoli cells. Our Leydig cells will be out kind of around in this top white area outside the tubule. Okay. And so we can see the generation and development of the sperm as we're moving forward. So we're not starting with a fully mature sperm. We're not starting with something we would recognize head with an acrosome, a midpiece, and a tail. We're starting with just a kind of generic looking cell. And it's generic in a couple of ways, but one of those is that it's still a diploid cell. So it still has two copies of each of your chromosomes. We still have homologous in these spermatogonia that we see up at the top here of the Sertoli cells. All right. So those spermatogonia born with a certain number of them, but that doesn't really matter in males because the spermatogonia are capable of mitosis. So because they're capable of mitosis, mitosis is that process where we make an exact copy of the same cell, we can just keep making more and more and more spermatogonia and keep them up in this top kind of layer here at the top of the Sertoli cell in this kind of basement zone or this basal zone. Okay. So we can keep making more spermatogonia, but we're also going to take some of them and split them up so that we eventually get mature sperm that only have half the genetic information, that are haploid cells, which is how we end up with recombining different genetics during actual sexual reproduction, combining sperm cells and egg cells. Right. So a spermatogonium that is not doing mitosis is not just making identical copies of itself up top, is going to start to prepare for meiosis. Okay, so meiosis is where we divide the genetic information twice, basically, ending up with 4 cells, each of which has half of the genetic information, basically. So we're going from diploid to haploid, and we're pulling out homologous pairs. All right, so as we set up for that, we're moving a little bit down, moving towards the lumen of the seminiferous tubule. So you can see here the primary spermatocytes are going to undergo meiosis 1. So they're starting that meiosis process. Then we go into meiosis 2 because we have 2 cycles of division to end up with our eventual gametes, leading us to our spermatids, of which there are 4. So we took one cell, pulled it in half, went from diploid to haploid, then we pulled apart again each of those haploid cells to end up with these 4 spermatids. And then they start to gain additional structures, start to differentiate, produce those typical sperm parts that we could recognize: the head, the midpiece with all the mitochondria, that tail or flagellum that eventually will allow movement at the end here. So that's when they're becoming spermatozoa here. We're reviewing that because this is about where we left off. Okay. So that spermatogenesis process is happening from the basement membrane, so that kind of connective layer at the outside of the Sertoli cells, moving in towards the lumen of our tubule. Okay. So one spermatogonium is going to stay up top, keep making more and more and more spermatogonia so that we have really lifelong ability to produce sperm. This is why, because we are able to take that initial germline cell and continue to make identical copies, continue to do mitosis. Okay. But for the sperm that are maturing, that are developing, we're moving down towards the lumen as we become more and more sperm-like and as we undergo the 2 phases of meiosis, until we end up with these spermatozoa that are released into the lumen of the seminiferous tubules. So we end up with the spermatozoa that start to look like sperm that you would recognize as sperm. We're putting them in that hollow part of the tubule that we're looking at. Now, they're not actually ready to move yet. This isn't happening like on demand in a kind of like minutes to hours way. These spermatozoa that you're producing are going to stay immotile, so unable to move, for about 20 days. So we've made their parts, but they're not yet able to produce a bunch of ATP, do whip-like movements of that tail, have fully mature sperm qualities in a kind of behavioral way yet. So these spermatozoa are going to be chilling in the lumen of the seminiferous tubules until they actually exit the testes. Okay, but they can't move, right? So they have to get out of the testis through peristalsis. So peristalsis is that kind of waves of contraction. So we thought about waves of contraction when we're thinking about how you're moving food through your intestines, right? And ultimately out through the colon, out through the rectum, out through the anus, right? We move stuff through peristalsis by squeezing progressively anytime we have a tube, including these seminiferous tubules. So they also can have these progressive waves of contraction, this progressive squeezing that moves these immotile, kind of paralyzed spermatozoa out of the testis into the epididymis, okay? So there's peristalsis. We're also moving some of the luminal fluid along with it. So that fluid is also kind of dragging these spermatozoa along with it. And it's as they're exiting the testis and ultimately exiting the epididymis as well, that they gain the ability to move, that they acquire motility, acquire the ability to move those tails. Okay. But they're kind of new to it. So as they're going from the epididymis up and into the vas deferens that's lying within the spermatocord, a lot of that is also still happening by peristalsis. And that is where they're going to be hanging out as mostly mature spermatozoa until ejaculation happens. So we're going to move them out of the testes when they're ready. So they're hanging out in the testis for like 20 days, even after we have the right combination of genetic information there. We move them out of the testis into the epididymis through peristalsis, and then keep moving them a little bit into the vas deferens where they're going to wait. Okay. So we are next going to talk about erection, emission, and ejaculation. So I'm going to ask you, based on what you've read, whether you've looked ahead at the slides, or just based on knowing some things about the nervous system, what part of the nervous system do you think is going to be kicking off our sexual response in males here. So what part of the nervous system do you think is going to cause the erection of the male sexual organ, the penis? Ooh, okay, cool. So we got a variety of opinions here. So let's go through it. All right. All right. So we mostly got to the fact that the sexual response is going to be controlled by the autonomic branch of our nervous system, right? So we split up somatic, going to like skeletal muscle stuff, from autonomic, that involuntary part of the nervous system that does these automatic processes like smooth muscle, like your heart, right? So we got to the autonomic. Then in the autonomic nervous system, remember that we have both parasympathetic, our rest and digest portion, and sympathetic, that fight or flight branches. Okay. So both are eventually going to be involved in the sexual response in males. And there's a handy kind of, I'm not gonna call it a mnemonic, but whatever, a way to remember which part goes with which phase that I did not come up with. So please don't get me in trouble, but I'm going to tell you because it's super useful, which is a point for parasympathetic and shoot for sympathetic. We're going to see that the erection is the responsibility of the parasympathetic nervous system. So as the penis becomes erect, that's your parasympathetic nerves doing that. We're going to have a phase called emission where we start to move some fluids. Then ejaculation, the actual exit of sperm out of the penis, out of the male copulatory organ, is going to happen through the action of the sympathetic nervous system. So both branches of the autonomic nervous system are eventually involved here, but it's the parasympathetic nerves that are going to start us out. Okay. So this is mostly going to be happening through spinal reflexes as well. We'll have some input from your brain in some of the early, contributing to some of the arousal phases, but a lot of this is kind of more local, more automatic. Okay. So we're going to go through the erection phase, the emission phase as we start to have fluid moving, and then the ejaculation phase as we actually have semen leaving the copulatory organ. Okay. So in terms of what type of receptors, like physiologically, are kicking this off, we're going to be thinking, all right, like just like in our digestive system, we can have thoughts that are involved in this process, right? So we had cephalic phase impulses in our digestive system. We do have descending signals from higher brain centers that can act as stimuli for arousal and for each of these phases. But we're also going to have mechanoreceptors, so physically receptors on cells in the penis and in other erogenous zones that also lead to these different phases of the sexual response. Okay, so we're going to look at this little flow chart next. Okay. Yeah. All right. So what we're going to be looking at is to get to our spinal cord, which is what's going to be sending the signals directly to the tissues that we're interested in. We're going to have both that mechanical stimulation of receptors, usually in the penis, and those descending neural pathways from the brain, from any thoughts, sights, whatever, coming to the spinal cord. Okay. So what that's going to do in the spinal cord is we're going to start to toggle the activity of our sympathetic and parasympathetic nervous systems now. Okay. So for erection to occur, what's happening with these stimuli for arousal is they're going to start suppressing sympathetic activity, and they're going to start raising up the parasympathetic innervation to the penis. Okay. And this matters because the change that happens is going to be in the blood vessels. So the way that erection happens is basically that we're trapping blood flow in the penis, right? So usually throughout the body, we've thought about the sympathetic nervous system and its effect on like smooth muscle, especially in places like arterioles. We talked about it when we talked about mean arterial pressure. We don't usually think about the parasympathetic nervous system as being particularly involved with smooth muscle related to blood vessels, but here in the penis, as we're thinking about arousal, this is a special case, it is. So as we're raising that parasympathetic portion of our innervation, so that parasympathetic response, what we're going to be doing is relaxing smooth muscle in blood vessels in the penis. So as we relax the smooth muscle, right? So we're making arterioles wider, we're making them dilate. So we're getting more blood flow, right? This is what the parasympathetic branch of the nervous system is doing here. Okay. So these blood vessels are going to be running through the erectile tissues in the penis. So those are the ones that ultimately swell up and make that physical structure stiff. So we're increasing the blood flow, which means we're increasing the amount of blood that is in those erectile tissues. So we're increasing the blood volume, we're increasing the blood pressure, all in this area. Okay. So that's one thing already, right? So we already have more blood. So that begins this process of erection. As those tissues start to swell because of this increase in blood flow, blood volume, blood pressure, they're going to impact the veins next to them, right? So blood needs to go somewhere and it needs to come back. But in this erection process, we have so much blood going to these erectile tissues in the penis that those floppy walled veins start to get compressed down, they close up, and then the blood can't return to the heart. So this is one of the places that we actually do think about positive feedback happening in the body. Usually when we see these flow charts, it's almost always negative feedback loops, but I'm going to point out, right, this is a positive feedback loop we see here, right? So this parasympathetic innervation, relaxing the smooth muscle, increasing blood flow is then going to start compressing the veins. And that means we're going to have more and more of that blood volume staying in the penis, which means that blood pressure in the penis is going to be going up and up and up and up. And it's literally that pressure of blood that is causing the erection. So that filling with blood is erection, essentially. Okay. So the main other phase that you probably think about in normal life is ejaculation. That's going to be specifically as we see semen leaving the penis. Emission is kind of the in-between part. So as we are moving from this phase where the penis is becoming erect, we also need to start moving fluids forward, right? So we've had the spermatozoa that we made in the testes. They're kind of hanging out in the vas deferens, but in order for ejaculation to eventually occur, we're going to need to move all those things kind of up and out, and they've got a relatively long way to go. So emission is this phase where we start to have contractions in these structures in the male reproductive tract, moving those spermatozoa forward and starting to have that seminal fluid be produced, adding the contributions of our different glands, moving all of this towards the ultimately exiting the penis as semen. Okay, so this is happening as we continue mechanical stimulation, those mechanoreceptors in the penis in particular, right? Our neural activity once we achieve erection. So during this phase of emission, where we're going from erection towards ejaculation, this is where we see our shift from the parasympathetic nervous system, which was relaxing your blood vessels so that blood is going there, right? Now we're going to switch over to sympathetic control, which is what starts to move semen forward and ultimately is going to cause it to be released. So this switch over from parasympathetic to sympathetic is happening during this phase. Okay. So during this phase, we're going to see contractions of the epididymis, the vas deferens, and the ejaculatory duct. So the different parts of the reproductive tract as we're getting towards exiting the body. This is when we're going to see the secretions from the seminal vesicles and the prostate going into this ductwork as well, right? So the vas deferens coming up and into the body through the inguinal canal, heading behind, and then through the prostate is getting that contribution from the seminal vesicle into the ejaculatory duct. And then they continue together and enter into the urethra, right? So ultimately, during this emission phase, this kind of in between, is when we have the sperm combining with the fluid from those glands, heading towards and into the urethra. In ejaculation is when we have it actually released out of the body. So we've removed all the stuff, the sperm, the fluids into the urethra. They're not out yet. During ejaculation, they're going to exit. So this is happening through the action of the sympathetic branch of the autonomic nervous system. So for these phases to continue, we're going to expect all those stimuli that initially started the whole sexual response process to be continuing. So this is still a response to mechanical stimulation, continued stimulation of these pathways. Okay. What's happening here when the sympathetic nervous system gets involved and leads us to ejaculation is we're moving from an erection, relaxing stuff and having blood flow in. So we're going to start contracting smooth muscle, right? So now we're not relaxing smooth muscle, we're contracting it. And we're going to be contracting it in specific places. So that's going to be happening in the urethra itself. So that passageway out of the body through the penis, as well as in skeletal muscle, actually. So we're going to get some kind of spillover nervous activity in some skeletal muscle at the base of the penis. An important thing that's going to happen during this process is we basically have a common duct, right? The urethra is a common tube for both urine and semen. So it's important that during ejaculation, one of the things that's happening here as the sympathetic nervous system is getting involved is it's closing the urethral sphincter, that internal one by the bladder, right? So it's closing off the movement of urine into the urethra so that we just have semen, those sperm, and all those fluids that we're putting in with the glands coming through the urethra during the ejaculation phase. So our ultimate result here in ejaculation is as we have these contractions of smooth muscle, they're ultimately going to be moving semen up and out through the urethra and out of the body. And that's what ejaculation is. Okay, cool. We're now going to move into a comparison. So we're going to keep those things in mind, and we'll loop back at the end and try and tie all these things together. But right now we're going to switch over and think about the female reproductive system and how that's a little bit the same and a little bit different. Okay. So when we're thinking about the female reproductive system, we're going to have a lot of cell types that are analogous. We already know that we have a reproductive tract that has developed kind of in parallel, like physically parallel to the precursors to the male reproductive tract. And we know it continued to develop because we didn't have an functional SRY gene producing testis determining factor, which didn't produce any testes, and those testes didn't produce any testosterone, right? So whatever didn't happen, none of that happened. So we kept our ducts going, kept maturing them into this female reproductive system. Okay. So some notable features of the female reproductive system. One is that we have cyclical changes in how this reproductive system functions. So when we look at what hormones are involved and what are they doing, we're going to want to be thinking about where within the menstrual cycle are we? That's something that you want to keep an eye to, make sure to read like figure captions, because otherwise it can get a little confusing. We're going to see that the female reproductive system, because of these cyclical changes, has restricted periods of fertility, meaning sexual reproduction can't happen at any time. It's not any time, any day of the week, any day of the month that we could produce a zygote, an embryo, a fetus. It's only going to be when we have ovulation occurring and we have basically an egg ready to be fertilized that is released, that we're going to see even the possibility of fertilization occurring. That's different than in our males, right? Because in males, we can see that happening any day. And in the female reproductive system, we have a limited ability to produce gametes. So we have a pool established at birth of gametes. And once we run out, we're done. So we're not going to see this continual production like we do when we're thinking about sperm. Okay. So thinking back to before break, I mentioned we had our female reproductive tract developing alongside the male reproductive tract until we get hormonal signals telling us which one to keep. So which reproductive tract was that? Look. Yeah. So the duct that we're thinking about when we're thinking about the female reproductive tract in development is the Müllerian duct. So this is what is going to develop in absence of appropriate signals, right? So we start out, Müllerian duct is the baseline. We have a little Wolffian duct developing alongside it. If we go towards the male reproductive tract, we needed testosterone to tell the Wolffian tract to continue developing. And we needed our MIH, the Müllerian inhibiting hormone, to turn off this development of the Müllerian duct. Because if we get no signal of any kind, your body's going to say, great, we're producing a female reproductive tract, Müllerian duct, go. Okay. So that's the pathway that we're working with as we're going towards this female reproductive system. Okay. So when we're thinking about what cells and tissues we're looking at, we're going to be looking in at the ovary here. So the ovary is going to be analogous or homologous to the testes, right? So this is what's going to be producing our gametes, okay? And if we zoom in, within that ovary, we can see a bunch of little dots, right? So what we have developing here inside the ovary are referred to as follicles. Okay, so each of these little follicles is going to contain one ovum. So that's what we colloquially would be referring to when we talk about egg cells, right? So we're going to have ova inside these follicles. And throughout the menstrual cycle, we're going to see these follicles, which are the ovum and surrounding cells, so surrounding structures, growing in size, developing, and then one, usually one, hopefully one, normally one, is going to win out and start to suppress all the others, which are then going to degenerate. So that's why you see different follicles, little different dots of different sizes here in this image here. Okay. So these follicles contain an ovum. They're developing in the ovary. And they're going to start out as what we refer to as primordial follicles, right? So our primordial follicle is going to be the smallest one, the ones that are just starting out, okay? So if we look at a primordial follicle and zoom in, we're going to pick one of these small little dots here. And we're going to see that it has an ovum or an oocyte in the center. It also has associated cells around it. Here they're labeled as the follicle cells. We're going to see that that's true, but we also have these cells called granulosa cells. As we walk through the menstrual cycle, we're going to see a lot of cell development, a lot of changing cell types, a lot of maturing cell types as we walk through. Okay. But if we're thinking about those granulosa cells, which at this point we're going to roughly think about as the cells surrounding that ovum, we're going to be thinking about them as being target cells for hormonal signals, basically, right? So when we're thinking about our testes, we're thinking about the Leydig cells as the ones producing testosterone. We were thinking about the Sertoli cells. One of their things was that they were the target cells for some hormones, right? So this is what the granulosa cells are going to be doing. So our granulosa cells are going to be receiving hormonal signals here. So they're going to be target cells for estrogen. So one of our hormones associated with the female reproductive system. And they're also going to be target cells for something called FSH, so follicle stimulating hormone. So we also see FSH in males when we're thinking about how they're producing sperm. So it's not specific only to females, but it's clearly named after these follicles in the ovary, right? So follicle stimulating hormone is going to be acting on these follicles in the ovary. Okay. So in response to estrogen and this hormone FSH, follicle stimulating hormone, the granulosa cells are going to secrete their own chemical messengers. So they're going to have a chemical response to receiving these signals. Okay. They're going to be doing a bunch of things, but one of the things that they will do is secrete something called inhibin. So when we think about inhibin, you're going to notice that it's really similar to the word inhibit, right? That's because, on purpose, this is a signal, a hormone that is going to be providing negative feedback. Okay. So when we're producing inhibin from these granulosa cells, that's going to be running back up the chain and suppressing some of our FSH secretion, some of our follicle stimulating hormone secretion. Okay. The granulosa cells are also producing some of the estrogens, right? So they're target cells of estrogens, but they're also producing estrogens. And they're going to be helping that developing egg cell as well through movement of nutrients. So they're surrounding that oocyte, they're surrounding that egg cell, that ovum, and they're going to be passing nutrients through gap junctions, so like pores in the cell, basically, as we're working on getting these different egg cells, these different follicles to grow. Okay. So now we're going to take a look and compare the development of these egg cells to the development of sperm. So you can see that we have a similar pathway, but we're going to have some key differences here. So in terms of similarity, right, you can see that we have cells splitting, right? So we're going to have cells that ultimately undergo 2 phases of meiosis. But what we're going to notice, if we pay attention to the words and the captions on these diagrams, is that a really important difference is that once you are past fetal life, right? So once someone with ovaries is born, that's it. They're done producing all the oogonia they will ever produce. So the spermatogonia undergo mitosis over and over and over and over again, make more and more and more identical copies. No like adult or even child is going to be doing that with oogonia. Okay. So we do have oogonia analogous to the spermatogonia, but you would find those in a fetus. So you would find those like in utero. So the diploid cells that lead to gametes, this is happening as we have the reproductive tract developing in utero. This is happening as gestation is going on. So during the course of a pregnancy. Okay. So that's when our mitosis and differentiation would be happening. And this is why we have this kind of limited fertility in terms of the lifespan in females, is because we only have a certain number of oogonia that are ready to keep going towards completing meiosis. And I say completing because it's not just that the oogonia can't do mitosis, can't make identical copies in an adult woman. It's that they have actually already kind of started meiosis. Okay. So we have a fixed number of oogonia, and meiosis begins during fetal life. So we set up for meiosis, and we end up in a phase of meiotic arrest. So where we've set up for meiosis, we've kind of sort of started it a little bit. We got to pause. And that pause is happening like when you are born, right? So you're already set up, ready to go through meiosis when you are born with these developing egg cells. And that meiosis just continues during our menstrual cycle and doesn't actually complete until after fertilization. So if we're thinking about the development of a sperm, we might be looking at, a little under a month if we're including that 20 days that they're hanging out unmoving in the seminiferous tubules. If we're thinking about how long it takes for one of these oogonia to get all the way from the initial mitosis through to the end of meiosis, that's going to be a couple decades, basically. Okay. So this is a big difference. So here you can see that the words for the different cell types do mirror what we saw in the spermatogonia. So our initial cells in fetal life that would have undergone mitosis to make more of them in utero would have been oogonia. So those would have been the diploid ones. And then we had our primary oocytes. They start to set themselves up for meiosis. So they're making their extra copies. And then they pause that meiosis 1 phase at birth, essentially. And they don't continue until just prior to ovulation to finish up meiosis 1, getting us to secondary oocytes. And it's not until after fertilization that we finish up meiosis 2, getting our fully developed ovum. Okay. So we've got the menstrual cycle, which is going to relate to the maturation of these primordial follicles into mature follicles and ovulation in the ovary. We're also going to see similar but different phases in the uterus. So there are going to be changes in the uterus as well over that month-ish long cycle as well. So we're going to be splitting up, thinking about our ovarian cycling, which are going to split into a follicular phase and a luteal phase. So we're going to split what the ovaries are doing roughly in half. And when we go to the uterus, we're also going to see changes in the uterus. We're going to split that up roughly into 3. So we'll take a look at that after we dig through the ovarian cycle for a while. Okay. Both of these subcycles, so the menstrual cycle overall and the ovarian cycle and the uterine cycle, are caused by changes in estrogen and progesterone concentrations. So they're not happening just because, they're happening in response to hormonal levels, which is why things like hormonal birth control work, right? Because these are a response to something happening in the body. Okay, so we're going to think about the ovarian cycle first. Okay. So when we're thinking about the ovarian cycle, we're focusing in on those ovaries, right? So the ovaries would be located in your pelvis at the end of your fallopian tubes, kind of hanging out there, kind of cupped a little bit by these processes from the fallopian tubes called the fimbriae. They're kind of like finger-like processes that are going to be ready to catch an egg cell at ovulation. Okay, so in that ovary, first half of the ovarian cycle, we're going to refer to as the follicular phase. So named for those follicles, right? So the follicular phase actually starts with the first day of a menstrual period. So the follicular phase is the days that someone's period is happening, right? So the menstrual period, followed by another about a week or so until we get to ovulation. So in total, we have about 14 days that line up with the follicular phase. Our dividing line is ovulation. So ovulation is when you can see we are actually going to be expelling a cell or collection of cells from our ovary. So you can see this kind of explosion happening here. That's ovulation. Okay, so ovulation kicks you into the luteal phase. So the luteal phase is the second half of the menstrual cycle. So our egg cell has left the ovary, but we do still have structures growing and changing inside the ovary that we're interested in. So we can see this over here. So that's about another 14 days in the luteal phase until we enter menstruation again. So until someone's period starts, which loops you back up to the follicular phase. So in total, the cycle will be about 28 days. That's our kind of textbook benchmark for this, 14, then 14. So just cutting it roughly in half. There is a lot of variability from person to person, from month to month, in how long an actual menstrual cycle would last. So this can be a little bit unpredictable. Okay. So let's think about that follicular phase in depth a little more. Okay. So we're starting out with our primordial follicle. So it's getting ready to maybe get towards ovulation. We've got our ovum, we've got our granulosa cells around it receiving signals, okay? So we're gonna have our cells in the ovary itself that produce estrogen, responding to estrogen as well. And we also have FSH coming from other parts of the body. So follicle stimulating hormone is coming from outside the ovary. FSH and estrogens are working together to stimulate our follicles to grow and develop. So they're going to be getting larger, and we're going to start to see more cell types kind of collecting themselves in that follicle. So we start out with a couple of primordial follicles that start to get bigger and bigger. We're not going to pick just one. We're going to affect a couple of them, a handful of them, and eventually we'll have a winner, but we don't know that yet at the beginning of the follicular phase. Okay. So our oocyte is growing inside this follicle that's developing, and the granulosa cells that were surrounding it are proliferating, so we're making more of them. And we're going to start to form some new zones in these growing follicles made up of some new cell types. So we're going to have a new region that we refer to as the zona pellucida, and we're going to have a region we refer to as the antrum forming here around the oocyte and within those granulosa cells. We'll take a more detailed look at a picture or with labels in a little bit. Okay. But we're going to have multiple follicles continuing to develop during these early parts of the follicular phase. Okay. Partway through the follicular phase, one of those follicles is going to be obviously bigger and more mature than the others, and it's going to become the dominant follicle. So it's basically going to win, and it's going to start sending signals out that end up suppressing all those other follicles that were also kind of in the race trying to become the dominant follicle. And so those other follicles that have been growing are going to regress, they're going to degenerate, they're going to go away. They're going to basically get reabsorbed. So it's only the dominant follicle that's going to keep going. We're going to have more process is continuing. That dominant follicle, after it suppresses the other follicles, still isn't done. It's going to continue to develop. It's going to produce a new zone called the corona radiata, and it's going to become a fully mature follicle called a Graafian follicle before we hit ovulation. We're going to run through all this stuff in a couple of different ways. So I know that was a lot of words, but don't freak out. Okay. So a couple more things that we're going to be piecing together as we start to get a handle on the ovarian cycle. We got a couple more hormones that we need to think about. Okay, so during the follicular phase, in addition to thinking about estrogen and FSH, we're going to be thinking about gonadotropin releasing hormone, GnRH, and a hormone called luteinizing hormone, or LH. So in order for ovulation to occur, so in order to have our mature follicle, our Graafian follicle burst and actually send our egg cell into the reproductive tract, we are going to see a spike in hormonal levels, essentially. So we refer to that as the LH surge. So we're going to see a surge in this hormone, luteinizing hormone, that triggers ovulation and sends us into the luteal phase. That's where it gets its name from. Luteinizing hormone sends us into the luteal phase, and we're going to see that we refer to this thing called the corpus luteum when we're in the luteal phase. GnRH is a hormone that's coming from your pituitary that's causing that LH surge. So that's why it's also got to be in the mix as we're getting a handle on these pathways. So we're doing some like word definition up front here. Okay. After ovulation, when we go into the luteal phase, we're going to be left with the remains of our follicle, right? So we're going to release our oocyte into the female reproductive tract. This is going to get caught by some fimbriae, going to enter the fallopian tubes, head off to the uterus. But we still have the rest of the follicle sitting in the ovary. So that ruptured follicle in the second half of the menstrual cycle in the luteal phase is going to turn into a gland. So it's going to change what it's doing. It's going to turn into a gland. That gland is called the corpus luteum. Okay. So that's what this thing is in yellow. I think luteal actually might mean yellow, but I'd have to look that up for you. Okay. So the corpus luteum, as it becomes a gland, is going to take over the production of some hormones. So it's going to take over production of estrogen, and it's also going to secrete another hormone associated with the female reproductive system called progesterone. So that corpus luteum, that ruptured follicle, is making estrogens and progesterone. And it's going to do that for a while. So it's going to be working, producing estrogen and progesterone for about 10 days. Then it's going to start to degenerate itself. So eventually it dies off as well in a process called luteolysis. Okay. So as luteolysis happens, that's going to see a reduction in the hormones that the corpus luteum was producing. So as it's dying off, it's not going to be making as much estrogens and it's not going to be making as much progesterone. And that withdrawal of hormones is actually what's going to end up triggering menstruation. So that's what's going to trigger a period. It's flipping us back from the luteal phase, ending the luteal phase, sending us back to the follicular phase. That's what happens if fertilization doesn't occur. A different pathway would happen if that oocyte that we released in ovulation around day 14, sent into the reproductive tract, if it does get fertilized, we have a whole different set of signals that go out to the body. And this gland is going to stick around. We're not going to see this gland degenerating. And in fact, it's going to kind of work in conjunction with some of the developing cells associated with the growing zygote, embryo, and fetus as gestation continues. Okay, so we'll pick up with this next time, which is perfect because I was going to review all those words we just said in a different picture. So we'll start off on Friday with that.